Hello, hello! Welcome to the 5th episode of Entire Ocean podcast. My name is Aya, and today I'm presenting my research work that I had done in Italy for my master thesis. It is the characterization of photosynthesis and carbon allocation in marine microalgal species acclimated to different light regimes. So marine microalgae carry out photosynthesis that takes place through two reactions. The first one comprises in a series of light reactions that occur in the thylakoid membranes found in chloroplasts. Two water molecules are split using light energy into oxygen, protons, and electrons by the photosystem II that is associated to light harvesting complexes. This activates an electron transport chain throughout the thylakoid membrane, at the end of which reducing power or NADPH is produced by the photosystem I. In parallel, the buildup of protons on the luminal side of the thylakoid membrane activates the ATP synthase that produces an energy molecule known as ATP. These two molecules are then used in the so-called dark or Calvin-Benson cycle reactions to reduce CO2 to carbohydrates. Think, for example, sugars. But not only, they are also used in other metabolic pathways for the synthesis of, for example, proteins and lipids. So here's a question. What are the effects on photosynthesis and allocation of carbon into macromolecular pools as a function of different growth light intensity? Are they species specific? To investigate this, the research involved Tetraselmis suecica, green microalga that is classified under the family of Chlorodendrophyceae and is characterized by a rigid extracellular wall. Another green alga employed in the research was the Dunaliella salina, that is chlorophycean and has no wall, but can tolerate high osmotic stress due to the possession of a glycoproteic cellular coat. Both species belong to the phylum of Chlorophyta and have evolved their plastids through primary endosymbiosis, while the third species has secondary plastids that were acquired through secondary endosymbiosis of a red alga, and belongs to the vast phylum of Bacillariophyta. I'm speaking about Hyodactylum tricornutum, that is a pennate diatom. It is characterized by a poorly silicified frustule that has only one valve instead of two. So these algal species were grown under control, low light, and photoperiod conditions. Cultures were counted daily to obtain the cellular density within each flask. They were then sampled in the mid-exponential growth phase in order to quantify the pigments, proteins, carbon and nitrogen, as well as determine the macromolecular pool ratios and measure the in vivo photosynthetic performance. Growth was therefore tracked throughout a time course of 11 days, at the end of which it was observed that algae acclimated to low light and photoperiod grew slower than in control. Especially under low light, the cellular density upon entering into the stationary growth phase was lower than in the rest of the conditions. The maximum growth rate was calculated from the exponential growth phase and resulted the highest in control cells of Tetraselmis suecica, which was also observed in the other two species. Contrary to a more pronounced growth in control, all three species accumulated much more chlorophyll A and accessory photosynthetic chlorophylls, like chlorophyll B in the case of the two green algae and chlorophyll C in the case of the diatom, when acclimated to low light. Even though the ratio of chlorophyll A to accessory chlorophylls did not vary much between the control and low light of Tetraselmis suecica and Hyodactylum tricornutum, it was not the case for Dunaliella salina, who showed a significant decrease of the ratio under low light that perhaps occurred due to a change in the stoichiometry of the photosynthetic subunits. As for the photoperiod, the variable ratios were among species implied a species-specific response to this condition. The increase in the amount of photosynthetic chlorophylls, allowed an optimal photon capture in low light, clearly improving the photosynthetic efficiency. This parameter helps to assess the functioning of the photosystem too in capturing photons and converting them into chemical energy in a process known as photochemistry, and here it was measured right at the beginning of each in vivo photosynthetic analysis. To furtherly understand the physiological processes of photosynthesis, MPQ was measured. Now, this is quite a complex parameter that involves a series of molecular mechanisms that result in non-photochemical quenching of chlorophyll fluorescence. Generally, the main components of MPQ are fastly activated pH-dependent processes through which the exceeding energy is dissipated as heat. These components mainly depend on light-harvesting complex stress-related proteins in green algae and LHC-X proteins in diatoms, as well as on the reversible accumulation of specific carotenoids like zeaxanthin in green algae and diatoxanthin in diatoms. As seen on this graph over here, MPQ was close to zero at low illumination and increased till saturation at around 1000 microeinstein in Phaeodactylum tricornutum, showing the act of these fast mechanisms as also researched previously elsewhere. In Tetraselmis suecica, MPQ was induced much more linearly upon illumination, which can be seen either here on the left or on the right graph, where it also relaxed soon after switching the light off. And this suggested the act of the fast mechanisms that were also observed in Phaeodactylum tricornutum. Dunaliella salina, on the other hand, showed a much more complex contribution of MPQ components that might have included additional quenching mechanisms. On the right, in fact, we can see how the induction kinetics of Dunaliella salina had a completely different pattern than those of Tetraselmis suecica. In terms of protein cell quota, in both green algae acclimated to low light, the biomass and carbon concentration expressed per cell were lower, and so was the carbon to nitrogen ratio, which might have implied that the macromolecular pool storing nitrogen increased. As you may remember, the amount of photosynthetic chlorophylls increased in all species acclimated to low light, but here this, so did the protein content, suggesting that they must have preferentially invested the energy produced during photosynthesis into the synthesis of proteins that constitute the photosynthetic apparatus. This was additionally observed through the macromolecular pull ratios that were obtained through the Fourier transform infrared spectroscopy analysis, optimized for algal research in our laboratory by Professor Mario Giordano. This method allowed to portray the cell composition of microalgae through the assessment of vibrationally active functional groups of macromolecules, which can be seen from this spectra over here. So based on the macromolecular pull ratios and the previously mentioned absolute protein quantification, carbohydrates and lipids were semi-quantified and expressed in arbitrary units. Under low light and photoperiod, Dunaliella salina accumulated less carbohydrates than in control. In Tetraselmis suecica, a similar decrease was observed only under low light, implying that more proteins were produced at the expense of carbohydrates and a slower growth. Whereas Phaeodactylum tricornutum showed a substantially higher lipids accumulation, and despite the well-known fact that this diatom is an oleaginous species that prefers storing carbon into lipids rather than into carbohydrates, questions about why does Phaeodactylum tricornutum produce so much lipids under low light rather than under control or photoperiod condition remains to be answered. In conclusion, differences in acclimation responses were observed not only between the green and red evolutionary lineages, but were also observed between the algae of the same phylum. Tetraselmis suecica exhibits a more homeostatic behavior than Dunaliella salina that is seen through a relatively linear photosynthetic response, whereas in Dunaliella salina its regulation is much more complex. Additionally, along with the amount of chlorophyll A that changes under low light, Dunaliella salina also changes the stoichiometric composition of photosynthetic subunits. And finally, this is on one side reflected in a roughly constant macromolecular pool cell composition in Tetraselmis suecica, and on the other side in changes in carbohydrates and protein pools of Dunaliella salina. Thank you for the attention. I would also like to thank my university, Università Politecnica delle Marche, for providing me the right material and instruments for this research. And yeah, if you have any doubts, questions, comments, let me know. I'm available. So yeah, comment in the comment section below or contact us through our webpage, entireocean.com, and we will be more than happy to answer you. Thank you. Thank you for your attention again. Bye.